Okay, so picking up where we left off, the video is posted. You can watch the photosynthesis video that my slide's going to tell me not to forget, right? So these diagrams are really good for putting together the light reaction. We have an overview diagram for the whole process of photosynthesis. This is a good overview of the light reactions of photosynthesis. And it puts all of the players into their sequence so you can see where they happen. I understand that it's totally annoying that the first photosystem is called photosystem number 2 and that the second photosystem is called photosystem number 1, and everything about that is a pain. But if you think of them more in terms of players in this electron transport chain, maybe that will help you. So photosystem number 2, we're going to call in just a minute, let's make it blue, the water splitting photosystem. And so we're going to start with the photosystem that splits water. We're going to pass to an electron transport chain. The second photosystem is going to receive electrons from the first photosystem that have been passed down an electron transport chain. And our final electron acceptor is going to be NADP+ and make NADPH. Okay, so let's go through the photosystems and then we'll come back to this and then we'll talk about the light independent reactions. So photosystem number 2 is alternatively known as the water splitting photosystem or the P680 photosystem. And I'm just, as I was watching this video, thinking that maybe I ought to put water splitting in parentheses everywhere I have photosystem 2. Let's make a note. Because that's more about the process. And I really want you to understand what's happening, not memorize photosystem 2 and photosystem 1, right? So photosystem 2 is called the water splitting photosystem or the P680 photosystem. And P680 is even more esoteric in some ways because I really don't care that you memorize this, but it refers to the fact that the center chlorophyll molecules in the photosystem absorb best at 680 nanometers wavelength. And so that's their optimal light absorption. And so that's where that name comes from. I don't care that you know that. I'm not going to ask you about P680, but it's in your diagrams. So rather than you just wondering what does that number mean, I'm telling you it has to do with the optimal absorption of light by that chlorophyll molecule. Okay. So in photosystem number 2, light energy is going to be absorbed by the chlorophyll molecules in the photosystem, right? The energy is going to hit the chlorophyll molecules. The chlorophyll molecules are going to pass that energy from one chlorophyll molecule to another and ultimately to the center pair of chlorophyll molecules, which are the P680 chlorophylls, okay? In that process, those P680 chlorophylls are going to give up an electron and it's going to go up to the electron transport chain. And when it leaves and goes to the electron transport chain, that chlorophyll is missing an electron. And that's a really unstable state for it to be. It's so unstable that the need to replace that electron splits a water molecule. And so we actually use that transfer of energy to pull apart a water molecule. And we grab the electrons, me and my fellow chlorophyll molecules, grab those electrons that are released from water and use them to fill the need of the chlorophyll molecules. Okay. So photosystem number 2 is going to be constantly splitting water as it passes electrons to the electron transport chain. Every time it sends electrons to the electron transport chain, it has to split some water to replenish its need before it can send more. Right? So as it's receiving energy and those electrons are getting really unstable and they've got to leave, then we've got to fill by splitting some water. So this reaction is constantly splitting water. It's going to then, as a byproduct, produce oxygen because the hydrogen ions and the electrons go off to the photosystem and, sorry, I was like, why are you pointing at me? So the hydrogen ions and the electrons go into the solution and the oxygen is left over from that water molecule. So this is where the waste gas of photosynthesis, the oxygen that ultimately goes out to the atmosphere, comes from. It comes from the splitting of these water molecules. Okay? The electrons go into the electron transport chain. The hydrogen ions hang out in the solution. Okay? So they're going to be in the solution around the photosystem. That light splitting is going to replace the electron, produce our oxygen, and those hydrogen ions will ultimately, as the electron transport chain proceeds, be transported into the thylakoid compartment. Okay. It depends on where the electron transport chain is in the thylakoid. So where is that picture? Here. So if the electron transport chain is up here, some of the hydrogens might be actually on the outside, and they get pumped into the thylakoid compartment. If they're here, they're going to get moved into one or the other thylakoid compartment. The hydrogen ions get moved by the electron transport chain, and it all depends on how things are oriented there. The water molecules are everywhere. Okay, so if I think about this chloroplast here, remember that all of the empty space is stroma, right? And that the stroma is a thick fluid, but it's an aqueous solution. It's a water-based fluid. And so there's water molecules abundantly available in the stroma. This chloroplast is, again, I should make it green and square like a plant cell. This chloroplast, here's the nucleus. Let's just write DNA for nucleus. These chloroplasts are all suspended within cytoplasm, which is also an aqueous solution, right? So everywhere these reactions are occurring, water is always on hand. If you get to the point that water is not on hand, you are already in a really desperate situation for all of your systems, right? If you run out of water and that's the reason you can't do photosynthesis, then you're getting in a pretty severe state of physiological stress, close to death. Okay. All right. So because of that transport into the thylakoid compartment, we once again create concentration gradient. All the hydrogen ions are going into the thylakoid compartment, so we're getting a strong positive charge, voltage generating in the thylakoid compartment. Okay. Just like before, we're going to have chemiosmosis. Before we talk about that, let's move on to photosystem 1, which is the energy harnessing photosystem, or the P700 photosystem. Again, 700 refers to the fact that the chlorophyll A molecules in the center of the photosystem have an optimal light absorption of 700 nanometers. That's the only reason that that number is used. Okay. Photosystem 1 is the energy harvesting photosystem. So if photosystem 2 splits water, photosystem 1 harnesses the energy, and that happens via sending it off to an electron transport, right? Sending it off to NADPH. So here in photosystem 1, our second in the chain, we still have photons hitting the photosystem, right? This photosystem is also receiving light energy. And as that happens, energy is being passed to the P700 chlorophylls, and the electrons get excited, and one gets transferred up to our second electron transport chain. When that happens, just like before, the electron has to be replaced. But now we have an electron coming from the previous photosystem, coming down an electron transport chain. So it just is used to replace the missing electron. It's a lower energy state electron at this point, but it's fine. It gets added to our chlorophyll. Okay. So our electron arrives from electron transport chain 2, and the electron from the electron transport chain from the chlorophyll A molecule is going to get passed to one little electron carrier shuttle and then over to NADP+. And NADP+ is going to pick up that electron just like our FADH2 and NADH from cellular respiration and carry it to another process. So it's going to take that electron away to the next reaction that happens out in the stroma. Okay. So let's put all of this back together. This is a different diagram. And this diagram sort of shows you our context. This is a thylakoid that is at the top of the granum. We've got open stroma above. So this space out here is all the stroma. And then in here, we have the thylakoid space. And then this is the membrane that's creating that disc of the thylakoid, right? That context all makes sense? Okay. So light energy hits photosystem 2, our water splitting photosystem. Our pair of P680 chlorophyll molecules gets so energized by all those photons of light that it gives up an electron to a primary electron acceptor that gets passed down an electron transport chain. And as that happens, energy from that electron is used to pump hydrogen ions through and into the thylakoid space. Okay. Hydrogen ions that happen to be in the stroma get pumped through. Right? The need to replace these electrons is so strong that a molecule of water gets split. The electron goes to the electron transport chain. The hydrogen ions get added to the building concentration gradient in the thylakoid space. And oxygen is released into the thylakoid space. Okay. Light continues to hit our photosystem number 1, our energy harvesting photosystem. And as that happens, this P700 pair of chlorophyll molecules loses an electron that goes up to another electron transport chain. It gets briefly passed and then directed over to NADP+ to make NADPH. And the electron is filled from this electron transport chain. That need is met. Okay. Now, NADPH is going to leave. It's going to carry out into the stroma where the Calvin cycle is going to happen. So that energy product is going on to our next reaction that we're going to talk about in a minute. But we also need, for this reaction to carry out, we need some ATP. And ATP is going to be made by a molecule that is very, very similar to our ATP synthase that we had in cellular respiration. It's practically identical. It has the same name. The ATP synthase molecule here is going to allow hydrogen ions to flow through it, and it is going to synthesize ATP using the energy from the concentration gradient. So our concentration gradient in the thylakoid space is being produced by the splitting of water and by the pumping of hydrogen ions in from the stroma, right? We then let those hydrogen ions flow back out through ATP synthase, and we make ATP directly into the stroma. Those hydrogen ions can then be pumped back through by another electron transport chain to build up the concentration gradient again. Does this make sense? A little bit? Yeah, so we're going to call it photophosphorylation. Yep. So this is still chemiosmosis. So we had substrate level phosphorylation in cellular respiration, and that was when we were using an enzyme that was happening. And then we talked about ATP synthase carrying out chemiosmosis, the flow of ions through the molecule. That's still happening. This is still a chemiosmotic process. But we call this ATP production here photophosphorylation, ION, phosphorylation, because we're ultimately making ATP from light energy. Okay. So again, we're just transferring a phosphate group, and the photo is referring to the fact that the energy to do so comes from light. That may even be. So I know you guys hate this answer. It's the only answer I got. It's out in the solution. It's just there. And ultimately, at some point, plant cells and animal cells have to make new materials, right? And there's a whole other process to build nucleotides and to, you know, make those things happen. But we also are constantly recycling them. So this process that we're going to go to next, the Calvin cycle, occurs in the stroma, and it uses ATP. And every time it uses an ATP, it sends ADP and phosphate back to the stroma. And then this energy takes that ADP and phosphate and turns it into ATP, which goes to the Calvin cycle, where it gets broken down. It's constantly being recycled back and forth, right? And at some point, some of those molecules might become damaged or might have to be recycled in another way, and we may have to build new ones, but there's a cycle here of flowing back and forth in the stroma. Okay, so I already said this, but I have it formally on a slide. This ATP synthase is still chemiosmosis. We're going to use the potential energy from a concentration gradient and use it to power ATP synthesis. And so I put in this diagram because I like this to give you an overall picture. This is the stroma out here. And then this is the thylakoid lumen in here. And each of those dots is a hydrogen ion. Right? So we have plenty of hydrogen ions out in the stroma. We just have a higher concentration of hydrogen ions in the thylakoid space because we're creating that concentration gradient. So we're going to have a slightly lower pH in our thylakoid space, and we have this buildup of charge. When we let it flow through ATP synthase to come back closer towards equilibrium as those hydrogen ions try to balance, we're going to harness all that energy to make ATP. Okay? This is a chemiosmotic process, just like it was in cellular respiration. And we call it photophosphorylation, which I had just written down, right? Photophosphorylation is the light-driven chemiosmotic process, production of ATP. I'm only asking you to know these words because the source of the energy is different in these processes we talk about, right? In substrate level phosphorylation, the reaction happens because of an enzyme. In photophosphorylation, the reaction can happen because we have light energy. Okay. All right, this is yet another diagram of this process. Pick which one you like the best and use it to study. The beauty of using the OpenStax book is that I can pilfer all of the diagrams from all the books I've ever taught out of and present them to you so that you can pick which one you like best. I find that some of these diagrams help me understand things better than others and resonate with me more, and it's not always the same diagram at any given moment in time. So I give you the menu to choose the ones that help you the best. But just trace the pathway and try to visualize it. Use that video to try to reinforce it. We are going to go over the Calvin cycle. That's what we're going to do next. So foreshadowing. Are you like done with the light reactions at this point? Like, come on, move on. All right, 7.3, using light energy to make organic molecules, otherwise known as the Calvin cycle, or the light independent reactions. So let's come back to our overview. And this is sort of putting together what Riley was just asking about and thinking about what's happening in the chloroplasts and in the photosystems inside the thylakoids relates to what's happening out in the stroma. Okay, so this picture again, this is a chloroplast. Whoops, don't mind my writing. Here we have our thylakoids stacked up in a grana, right? I guess I wanted there to be 3 there. And the light reactions that we just talked about are producing ATP and NADPH, right? They're taking light energy and they're using that light energy to make ATP and NADPH, right? We haven't made any sugars yet. And ultimately, the goal, the end product of photosynthesis is some sugars, right? And we're going to do that in the Calvin cycle. The Calvin cycle occurs in the stroma. So the ATP from the light reactions and the NADPH from the light reactions are going to go out to the stroma where they're going to carry out the next step. And we call this next step the Calvin cycle. We also talk about it as the light independent reactions. They used to be called the dark reactions, but that's not really true. So light independent or Calvin cycle, either one is going to be where we're going to make the sugar. And these reactions will only occur as long as ATP and NADPH are available in the stroma. So as they occur and they use up ATP and NADPH, if new ATP and NADPH are not being created by the light reactions, then those reactions run out of steam and shut down. Okay? So they require energy from ATP and NADPH for the Calvin cycle, and ultimately our output is going to be G3P. Does anybody know what G3P is? Does that sound at all familiar? Have you heard G3P before? Everybody's saying no. Huh? It is the molecule that we had between the energy investment phase and the energy harvesting phase of glycolysis. So when our glucose molecule goes through those first energy investment steps in glycolysis, it splits in half. Right there, it's 2 G3P molecules. Okay, so it's half a glucose. The G3P is half of a glucose, essentially. So we need 2 G3Ps to make glucose. We generally talk about the outputs of the Calvin cycle as being glucose or sugar, right? The actual molecule that comes out is a G3P. And we need to have more than one Calvin cycle to get enough G3P to make a glucose, okay? But we still say our overall take-home product, what we really make, is glucose, because we're going to assemble those G3Ps.
So for the first part, is light stored like days?
It's a great question. Really, plants can't store light. They can, but they can store the products of light. So during the day, when the sun is shining and the plant's leaves are just basking in sunlight, they're cranking the light reactions, and they're just pounding out ATP and NADPH, and they're storing it in their stroma. And they're doing some Calvin cycle and making sugars. As sun sets, they can continue doing some Calvin cycle as they're using up those remaining materials. Generally, what we see in most of the plant species that we're sort of thinking of commonly all the time, the ones we're familiar with, is that photosynthesis, both the light reactions and the Calvin cycle, are daytime activities, and cellular respiration is a nighttime activity. And so when it gets dark, instead of trying to do this where we don't have our materials being made on a ready supply, we shift over to doing cellular respiration, and we burn our supply of extra glucose from the day and use it to carry out other activities like growth, like flowering, or working on producing seeds, reproduction, those kinds of things. But there's a balance. There's always a trade-off. Like you can't do all of your cellular respiration at night because you need to burn energy during the day too, right? But so the rate of photosynthesis to cellular respiration trades off depending on whether light's available. Okay. So the Calvin cycle, like the citric acid cycle, is another big cycle. Got lots of steps occurring. We're not going to worry about each individual step, nor am I going to ask you to know any of the intermediates. I like to think of the Calvin cycle as really 3 separate processes or 3 phases to the Calvin cycle. And this is the overview level at which I think we need to understand the Calvin cycle. So 3 things happen in the Calvin cycle. We fix carbon dioxide from the atmosphere. So we take that simple atmospheric gas and we add it to an organic molecule. Then we make G3P, which is our energy product. And then we regenerate the starting point so that the next Calvin cycle can proceed. Okay. So fixation of carbon dioxide begins with adding those carbons from carbon dioxide to a 5-carbon molecule known as RUBP. So RUBP is here left over from the previous Calvin cycle, and we are going to add carbon dioxide to it. And you'll notice in this diagram that we have 6 RUBPs, and we are going to add 6 carbon dioxides to get a total of 12 3PGs. Okay, so we need a lot of molecules for this reaction to happen. We're going to proceed through to synthesis of G3P and regeneration of RUBP as our starting point. I said I don't care about intermediates and all the enzymes that catalyze things, but I will mention Rubisco. The addition of this carbon dioxide to RUBP relies on Rubisco, which is the enzyme that catalyzes this reaction. Okay, it's worth noting primarily because Rubisco is the most abundant protein on the planet. It is hugely important. We would not have any food for any organism to eat if we didn't have Rubisco. So, most abundant protein on the planet. I don't know. I have no idea where Nabisco came from. You guys have great trivia questions, that's for sure. Okay, so we have fixed carbon, make some sugar, regenerate RUBP. So we start with our RUBP molecules, and we add our carbon dioxide. You'll notice that this diagram says 6. This diagram says 3. The discrepancy has to do with our output. The 6 is enough to make 1 glucose. So if we're doing net per glucose, we need 2 Calvin cycles. For each individual Calvin cycle, we start with 3 RUBPs, and we add 3 carbon dioxides, and we make 6 of our next intermediate PGA in order to proceed forward. This step, with the help of Rubisco, is carbon fixation. So we're taking a free-floating carbon atom from the atmosphere, and we are fixing it or adding it to an organic molecule. And so we're making a more complex molecule. Okay. Then we're going to add some energy. And we're not going to skimp on the energy because we're building something complex here. This is a strongly endergonic reaction. We've got to pour energy on these reactants in order to get our complex product that we want. So we're going to use up 6 ATP and 6 NADPHs. And again, every time you see the products of these reactions, these guys are just going back to the stroma. So we're regenerating empty electron carriers to go to the light reactions, and we're leaving behind ADP and phosphate that can be made into ATP again using photophosphorylation. Okay, they're just part of the cycle. This cycle continues forward, the Calvin cycle, and here, after we've added all that energy, we have some G3P molecules. We have a total of 6 for each round of the Calvin cycle, and those 6 molecules, 5 of them are going to move forward. Only 1 exits. So for each round of the Calvin cycle, we only get half of a glucose, 1 G3P. These 5 G3Ps all have to go on to our regeneration of our UBP. And that's going to take some more energy. So here we had our carbon fixation. That's not quite how I meant to draw that. Here we have our sugar production. And then up here we have our regenerate, our UBP. Your diagram does it much better than I do, but those are our 3 steps. Okay. This one G3P then can exit and be our take-home product. So to make one G3P, we have to do 1 turn of the Calvin cycle. We have to invest 3 carbon dioxides from the atmosphere, 9 ATP, and 6 NADPHs from the light reactions. Okay. We need 2 G3Ps if we want to make a glucose. So to make a glucose, we're going to have to repeat the Calvin cycle twice. Okay. So for every glucose molecule, we need 6 CO2s, 18 ATPs, and 12 NADPHs to make 1 glucose. You don't like cycles? You prefer a linear metabolic pathway? Everybody's got their favorite things. Okay. Once we have those G3Ps, we have lots of things we can do with them. If I need to just make stuff and burn energy in cellular respiration, there's no reason to make a glucose because I can bypass the energy investment phase of glycolysis and just shoot those G3Ps right into the middle of glycolysis, right? So some of those G3Ps are just going to go over to the mitochondria and be used to make more ATP for the plant, the plant's own metabolic processes. Other G3Ps can be joined together to make a glucose. Many glucoses can be joined together to make sucrose or to make a large starch molecule. We have an abundance of energy and the plant's just cranking out all these glucoses. It can make some starch and store it up, right? Plant wants to get bigger and make more leaves and grow. In order to produce more cells, it needs cellulose for its cell walls, right? So those glucoses can be used to build those structural components in the cellulose. There's lots of different potential fates for these molecules to take. Okay. Any questions about that? That's the end of photosynthesis. I feel really weird about wasting 10 minutes. You guys want to do some more clicker questions? Let's grab some more clicker questions. I feel like some of you are completely wasted and totally supposed to